All right, so let's go ahead and get started. Questions on the study guide. Yes.
But it's still kind of confusing because it's parts of it, the sarcomere, like, what is that exactly? Like, is it within the muscle fiber itself, or is it a bunch of muscle fibers together?
Yeah, so a sarcomere is the contractile unit inside a muscle cell.
Okay.
So it's like almost the most zoomed in you can get.
So it's like an organelle, essentially. Like,
It's a structure, yeah.
okay.
So like if you zoomed in, I got it actually up here. You start with the muscle and you keep zooming in, fascicule, muscle fiber. Myofibril is a whole bunch of sarcomeres.
Okay.
And then a sarcomere is the one little piece of actin and myosin.
The myofibril is what gives us the striations, right? Is that what, or is it
The sarcomere gives the striation. So if you put a bunch of sarcomeres together, end to end, you get a myofibril.
Okay.
And if you put a bunch of myofibrils together, then you get a cell.
Okay.
Can you explain the difference between the myosin and the actin? They just kind of, I kind of got the same difference.
Yeah, so the way to think of it is the actin is the piece that gets pulled on, and the myosin is the part that does the pulling. So the myosin does the pulling, the actin is what's getting pulled on. Yes?
Can you explain the isometric contraction?
Yeah, so the isometric versus isotonic, did anybody get those? Do you know which one's which?
Okay, the isotonic remains constant.
Yeah.
The isometric increase.
So which one involves like movement? Right? So like this is isotonic. Anything? If I'm just sitting here like this, that's isometric. But you can see I'm holding something, so there's still force. So it is a contraction, it's just nothing's moving. Right. So basically, yeah, pretty much always you're probably resisting gravity somehow. Isometric. So any contraction that doesn't produce movement is isometric. Any contraction where something moves, your fingers, your hands, your toes, that's isotonic. So I'm sure you've done any, exercising stuff. Isometric is usually the more painful kind. I think, like holding in a plank or, holding a 10-pound dumbbell out like this. Or like the shrug one. You've done that one. Anything else?
The A and the I bands.
So the A and the I bands. Show you a picture real quick, and we talk about them at the same time. All right. So the I band is where you only see actin. So they kind of split the sarcomere, into little sections. And we'll talk more about it, but basically the I band is where you only see actin. There's no overlapping with myosin. So you only see the yellow. And if you look at it in the microscope, you can kind of see it there. So the I band is the actin. The A band, if you notice, it's where the actin and the myosin overlap and where the myosin is. Because up here is yellow is actin and red is the myosin fibers. So the A band is where the myosin and the actin overlap and where only myosin is. The I band is where only actin is. right. Anything else? All good. Oh, I can put those things up too. Here you go. Let me get these out of your way. Yeah. Thank you. All right. Pass that one on up. Oops. All right, so the answers. Which of these binds oxygen? Is it sarcolemma, T-tubule, sarcoplasmic reticulum, or myoglobin? It's myoglobin. Pretty easy. Myo means part of muscle. Globin means oxygen. Not a function of muscle tissue. It is all of the above. So stabilized body positions generate heat. How does your skeletal muscle generate heat? So the shivering, definitely. And moving substances on the body is also one. And what property of muscle is related to its ability to stretch without being damaged? Is it excitability, contractility, extensibility, or elasticity? It is extensibility. So they're very related, but they're a little different. So extensibility is you can stretch without breaking. Elasticity is you go back. You get pulled back to original shape afterwards. So rubber bands, for example, do they have extensibility or elasticity? They have both, right? So you can pull them and they don't break, so they're extensible. And then if you let go, they go back, which makes them elastic or have elasticity. Just want to make sure you got those. And what's the motor that performs contraction? Is it actin, titin, myosin, or Z-disc? It is myosin. So you're asking me, what myosin was. It does the pulling, which makes it the motor that's pulling. And what type of muscle contraction does not involve any movement? Isometric, isotonic, isometric. So hopefully you got some of those. All right. 10. All right, so the reason this chapter is interesting and also hard is because you're going to learn exactly how, at the most tiniest molecular level, your muscles generate contraction. So instead of just saying, oh, your muscles contract and they get shorter, we get to actually learn how they do it and how it's controlled. So for part 1 today, we're basically just going to talk about what a sarcomere is, how it shortens, and then next time will be more about the signaling that controls contraction. So if you notice today, we don't talk anything about why it contracts or what tells it to. We're just going to stick with how it contracts for now. And that'll be basically the whole lesson. Of course, there's 3 kinds of muscle tissue: skeletal, cardiac, and smooth. Mostly just talk about skeletal muscle today. Cardiac muscle we won't talk about at all. Talk about that when we do the heart in 202, and just talk a little bit about smooth muscle. So of course, smooth muscle is the muscle that's involuntary, and you have it in your blood vessels, you have it in your intestines, it helps food movement. But mostly we'll talk about skeletal muscle. So which of these is not a function of the skeletal muscle? All right, so I can chat with your group there because we're tied between 2. All right, final vote. 54, 8, 12. Everybody vote? Just want to make sure they're all working. All right, interesting. So everybody changed to D there, which is the answer. So that's smooth muscle. All the rest of those are skeletal. Because are your eyes moving? I know you're kind of thinking that's involuntary, but it's not. You are telling your eyes to look one way or the other. It's just not all that conscious, right? You don't have to say, eyes, turn left. It's more like you're thinking, I wonder what's over there. And they just kind of automatically move. But you are telling them to. It's just not all that conscious. Right. So as you zoom in more and more, you have these different levels. So when you dissected the chicken, you saw a muscle, or the bicep, the tricep. So muscle's made of a lot of fascicles. Fascicles are made of lots of muscle fibers, which are cells. And then the cells are made of lots of myofibrils. Myofibrils are made of lots of sarcomeres. So that's your list from complex to simple as you zoom in and zoom in and zoom in. What would be smaller than a sarcomere, do you think? To what? Actually, a nucleus is probably a little bigger. What are sarcomeres made of? Yeah, so if you wanted to go even one step farther, you could go sarcomere and then myosin and actin, which are just proteins. But we'll get those too. And so for today, we're basically just going to talk about sarcomeres. So hopefully you can get the idea. Let me turn off one of these lights. All right, so just to give you an idea of what it looks like as you zoom in. There's your muscle, and it's composed of a bunch of sort of cylindrical tubes full of all the muscle proteins. Those are your fascicles. And if you zoom into those, you start to see individual muscle cells. So you see how we're just kind of zooming in. And then if you zoom into the cells, eventually you get to a myofibril. And then if we zoom in even more, inside those myofibrils, which are these, you could see the individual sarcomeres. So from one Z disc to another is a sarcomere. And so you can kind of think of it as almost like just a parallel bundle of ropes over and over again. And so, the muscle would be the whole rope. Each little strand, like that'd be the fascicle, and then each little string in there would maybe be a myofibril. And then if you just took sections of that string, that would be your sarcomeres. So we're going to talk about what's happening here. And if you notice, everything's kind of attached lengthwise. So what do you think happens if this sarcomere gets shorter? Or I guess I should say, what happens if all those sarcomeres get shorter to the muscle? Does it get shorter or longer? It gets shorter too. So everything's happening here, but you just multiply it, by 1,000,000, and suddenly you get the whole muscle shortening. And of course, like everything, it is in 3 dimensions. So it is like big circles or big cylinders of sarcomeres put together. All right, so I'll just go through these a little bit, and then you're going to be drawing it, because I know there's lots of crazy vocabulary and structures up there. So this is a diagram. I have one muscle cell. So you've got in blue the cell membrane, and if you notice, there's these special proteins here, membrane proteins. They're going to attach to the cell next to it. So it's like those desmosomes or adherence junctions that we did way back in chapter whatever. So each cell is very firmly anchored to the other so that they move. When one contracts, it just pulls on the other one next to it. They don't like separate from each other. So all the cells are attached together. And we'll zoom into our myofibril. So this is one cell. It has lots of myofibrils. And the myofibrils have lots of sarcomeres. So if you zoom in to a sarcomere, there's a bunch of sarcomeres. Here's one sarcomere. So the basic structure of a sarcomere, In the middle, there's an M line. These are structural proteins that myosin attaches to. So myosin's in red here. So it's tightly attached there, which is good. You need something to attach to on that side. And then on the two ends, we have what's called a Z disc. These are more structural proteins, and that's actually what you're going to pull on. Attached to the Z disc is yellow actin, and then in red here is your myosin. And so when we contract, this myosin actually pulls on the actin, kind of like on a rope. And we're going to do a demo of that in a little bit. And you've got all your different zones. H zone is where you only have myosin. I band is where you only have actin from here to here. The A band includes the myosin and the myosin actin overlap. So from here all the way over to there is your H zone. And then the last one, this titin here, if you notice it's attached all the way from the Z disc to the M line. So it's important for keeping that, letting it be extensible and elastic. It's that titin protein. Because if you can imagine, if you pulled your muscle, spread those Z discs apart, what would happen to the myosin actin overlap? Would it go away, get bigger? What do you think? Imagine you're taking these two Z things and pulling them left and right. What would happen to the amount of overlap between yellow and red there? It would decrease. And if it became too much, like if it became none, then the myosin can never pull on the actin. But because titin's elastic, if you pulled it apart, the titin's going to pull everything back together. Because it's like a little spring in there. And it's attached from Z to M. So if you pulled it, it's still attached, and you could pull everything back. So that's kind of how it works. All right. I had to fit a lot of instructions on this one. So basically the way I thought might work well for you to actually get an idea of what these sarcomeres look like is I've got step-by-step instructions for you to draw one on your table. So you're going to do it, let's see, all these instructions, in pairs, no books, and don't erase it because you're going to use it again in a little bit. So you'll need those markers, and I'll be walking around to make sure you're getting that. And if you have extra markers, let me know. Do you have any markers? What about in here?
No. I get my,
Do you have some extras?
extra papers.
Mm-hmm.
Do we only need one, or do we all need one?
Oh, one for each of you.
Okay, here you go.
I mean, one for, give one. There you go. There you go.
Thank you.
Sure. I'm gonna look around some more.
So, draw 2 parallel lines.
Oh, do your lines like this. Like one here and one down there.
What?
Like your first line's here and your second line's down by you. I wasn't sure how to write that.
So we're not doing like.
Well, because we're doing a sarcomere, we want it to be long. So these are our first two lines. So I put like one here, one here. I wasn't sure how to write it in the instructions. They're still parallel, but like parallel like this. If that makes sense. Okay. So you're good except just two of them.
Yeah.
Right? So this one and then that one way down there.
And then, so
And label them as you go too, so you know what you're.
Do we each need our own, or is there?
In pairs.
Okay. No, I mean papers.
Oh,
Do you have extra? Oh,
yeah. I don't know why I have so many.
But that one printed on 2 papers.
Oh,
Yeah. So you're good.
so I put a line here and a line down there.
I know, you can't really see. It's like you just drew
You might have an easier time doing it. Well, yeah. Why don't you do one here and then one here to start with? Because you need a lot of space. All right. How are y'all doing?
We're on C frame. Then 2 right here?
Oh, and you need to label them as you go, so you know what you're drawing. Okay, did you draw 2 of them?
This is the end line right here.
And that's,
The end line?
yeah, they call it a disc, or yeah, you can call it a Z line.
Z disc?
Okay.
This way or this way?
Well, did you do B yet? Really? I only see 2 lines. Oh, there it is. Sorry. It's covered up on my belly. I would label them as you go.
Oh, okay. So this is like. Wait, but then so do these see? Does that go this way or this way?
Well, if it goes,
The M line, does it go?
so draw 2 pair of lines that go halfway from the M line to the Z disc. So they're going to be perpendicular to those lines.
So maybe like this.
Well, that looks like it crosses the M line to me.
Oh, so it just has to go from the M line to the Z.
Well, read it one more time and see. But yeah, it does seem to be perpendicular. You're almost there. Is that halfway to the Z disc or all the way to it? Okay, you're getting close. You're getting very close.
Is Alma doing it right? I don't know. That's
So what does it say for C?
Draw 2 parallel lines that go halfway.
Okay.
So 2.
Well, is that line you're drawing going halfway from M to Z?
Yeah?
Really? It looks like it's going all the way to me.
It's because you put that back.
Oh, so it has to be like this.
So start at the M, go halfway to the Z. That's the M line.
Oh, yeah, yeah. And then the next one.
I'm done already?
So look accurate.
No. Did you go step by step on here? Or did you just try to draw it from memory?
No, we
Okay.
What part's wrong? All of it?
Quite a bit of it's wrong.
Draw the 3 parallel lines. That one's wrong, right?
parallel lines, draw line half.
So C was 2 parallel lines that go halfway from M to Z.
So the M3, but is that not right?
Well, that looks like it's going all the way from M to Z to me,
Oh.
right?
Which half? Which one do you start at? You start at
Well,
M and go halfway?
if it goes halfway from M to Z, let's start at M. So you're getting closer.
No.
Did y'all label everything?
Yeah.
Like thick and thin filaments?
Oh, no.
It's kind of tricky.
It is tricky. I messed this one up. So I messed up on what I wrote on there for one little thing. So for E, it should say Titan connects M line to Z disc. So I wrote it wrong on that paper. Oh, so I think what I'll do, so once you think you have it, look back at your book or the notes to see if you think you're right. And then I'll come check it. No, it doesn't mean it's wrong. It's just.
Sir?
Laughing at you probably now.
So like this. And then it connects like that.
They overlap, but they don't connect. It's like they go next to each other, just like that. So you want to label them too, like actin, myosin. But you're doing great. You need to label them.
No, we have to do the eighth round. And
No, like you only labeled the Z and the oh, sorry. But I want you to label them actin, myosin too.
Okay. Okay. I can do that. Can I have my marker back, man?
Oh, there you go.
So this is
And again, you should be doing this without any help. At least till you think you got it. And no, until you think you got it. Yeah. Because I know like, it's totally different, like reading it and putting it down than just copying it from somewhere.
Right,
It's a
right.
totally different thing to do.
Now we leave this on here?
Yeah. So you think you got it?
I think so.
So the only thing I messed up, the Titan needs to be fixed.
Doesn't it go here and then goes under?
Oh yeah. Okay.
Sorry, I just did.
Sorry, I was looking at that one.
Oh, no, I just did only this one.
That's my bad. I just wasn't paying attention when I wrote it.
There we go.
So then myosin, actin, zetas. Okay, this makes sense, right? Kind of.
The zones are the only thing that throw me off. But
Yeah.
I think so.
And we'll go over the zones too, but
Right.
It doesn't say, it doesn't specify, I guess. Draw two.
all right. So what do you think?
Well, I understand this more, but I'm really confused with that.
Yeah. So
Is that on every one?
yeah, it's all throughout, and it's basically like between the myosin and the actin, just from here to there,
Okay, so it's here, and then this would just be the thick, or the thin.
right? And just like you haven't had the thin and the thick sort of overlap
Yeah.
like that, that's perfect.
And so that's the tighten right here.
Instead of just these little coils?
Yeah, I mean,
I mean, we just put it like that because they're kind of like,
yeah. I don't really care how you draw it as long as you've got a Titan in there between them. That's kind of what they look like. Cool. So then you just have these 3 questions at the end.
Okay, check. How's that looking?
It's much better. The only thing you have left is your second piece of myosin and your Titan.
This is a tight.
No, that's your myosin. Right? You said that's myosin.
oh, did you label it?
She did. She's got it right. You've got it labeled right there. No, you have it right. It's not Titan. What you have is right.
Oh, okay.
So Titan connects M line to Z disc. So you'll need a new line for that one.
So I didn't do it like that.
You did it right,
Oh, okay.
right?
So like this?
Actin, myosin's
Oh,
here.
yeah.
Tighten
that?
over here, right?
Well, Titan would be like its own line, going all the way from here to there.
Like this?
Mhm. Yep. And then you just have a few other things to label. All right, how y'all doing?
How does that look?
Yeah, the only thing wrong, and I know it's because the book picture's a little confusing, the myosin actually stops here. It's not connected there like that. And I know the picture in the book kind of looks like that, but Titan actually does that. It connects the M line to the Z disc.
Oh, it goes all the way.
And it's sort of, you know, it doesn't really touch the myosin. Maybe it's here.
This says it connects with thick film.
I know I wrote it wrong. That's why I wrote it up there again.
So does this go
There's just how
Does it go
you're,
inside this line?
no, the Titan
Yeah.
just goes next to it.
Or
So
goes
it's
next
just
to
kind
it.
of.
Like above that.
Well, so it'd be like from here to here.
All the way.
All right, so where's your myosin here?
Right here.
Mm,
So maybe 2 actin.
so the myosin's like perpendicular to your M line.
I don't think so. I think that's what.
Would it be? It was supposed to be the ones that you deleted. The ones that you.
I would suggest just starting over real fast and just do one sarcomere. So there's one M line, there's a Z, there's a Z.
Okay.
Instead of trying to do like, because I think right now you're doing almost like 3 sarcomeres at once, which I think might be confusing you a little bit.
Okay.
We can draw.
I know
I
it's
mean,
crazy.
There's an H
And then
zone
the
or
A band
I zone or
is-
A band right there.
Yeah,
It'll be nicer on my paper.
yeah, I know. All right, how are y'all doing? So what are all these here? Does it seem like nothing's attached together to you? So for like C, for example, when it says go halfway from M line to Z disc, that means it starts at the M line and goes halfway. Does that make a little more sense? So that'd be a myosin. And then you could draw those, and then you could draw the actin.
Is this right? The I band was here. Titan.
So the I band, I think you could just see how you labeled the H and the A. I would label the I band the same way. So you know how they did it on the thing where they were like saying from here to here is the I band?
Right.
That's how I would do it. Because it's not, I'm not totally sure what the I band is the way you have it.
But the
Isn't it where it
up here.
overlaps?
It overlaps with here. I thought it was down here.
So the I band is where the actin is only by itself. So on yours, it would start here and go over.
Okay. Yeah.
Oh, so this.
And then this is the H zone. And then this is the I band again, correct?
Yeah.
Right.
Over here. Look.
And it's confusing, right? Because the H zone and the A band overlap each other. So the A band is bigger than the H zone,
Right, because the H zone is in the middle. Okay, I get it.
right? So the A band goes from, where does the A band start then? You're going to mark it on there.
Like that.
Yeah, the A band stops when you get to the end of the myosin. Yeah.
So did I do it right?
So basically, the A band includes all of the
So
myosin.
the myosin doesn't include the I band. The I band is the
Right.
actin. The A band is myosin only, which is in the middle.
The H zone is myosin only.
Okay.
No
Zone
actin.
is myosin over.
The A band is all of the myosin, even where it overlaps.
Okay.
And we've got a bunch of pictures of it too.
So kind of go like this. H zone is all the myosin.
band and here, and then the I band's right here.
Um,
They're overlapping.
no. So the I band, so if this is your A band ending, where your thick filament ends, that's where the I band starts.
I thought they overlapped.
No, the A band and the H band do.
And then I band is the
I think. Yeah.
I-bands are like, is that on the right spot?
Yeah, it's where there's only actin. Let me put that up here actually. Where was that one? How did I put it on there? Where's the more zoomed in one? If you want to check yours with up here, it might help, especially your I bands, H zones, A bands. Y'all getting that? Where's your Z discs? Did you cross them out? Oh, the Z discs, they're parallel to the M line. So like they're aligned here, which is what you had before, I think. At some point. That's a Z disc.
There we go.
And where's your myosin?
Yeah, she's like 36.
So where's your myosin?
Oh, hell.
It's drawn on there, but you didn't label it. Here. You got it?
I think so. Does this make sense? Because, because these don't actually touch, right?
Which one? The Titan?
The Titan and the A, do they actually touch or no?
Well, the A band is a location. It's not like a protein.
Yeah, but it's made of the
Myosin?
myosin, yeah. So, but it's just like,
Right.
gotcha. So, but these don't
How
actually
y'all doing?
Doing all right.
You're getting closer. Just try. That's all you do is try. One thing at a time.
I usually do my hair for
You just can't see his.
Just can't see it.
Here.
Oh.
Sorry.
Well, take a look up there. See if you think your A bands and
I think
H
I got
zones
it.
match
It
up.
just looks pretty attractive.
Kind of was listening to you talk to the other Titans too.
The only thing that needs to change on yours a little bit is the actin and the myosin should overlap a little bit. So like this is your myosin, right?
Yeah.
That's your actin? Right. So they do overlap.
Okay.
Otherwise they wouldn't be able to pull on,
Gotcha.
myosin wouldn't be able to pull on the actin.
So these two have to govern?
Yeah.
These have to be the control.
So they don't touch, they just kind of overlap, right?
That, right?
Yeah.
Right.
Yeah.
And that's, right, it's still attached.
Okay. That's pretty bad.
So the A band takes up all of the myosin,
So, that?
right? And the H zone is where there's no overlap, where it's just myosin,
I haven't drawn it just.
right?
You can't see it.
He's like, I can see it.
Right?
So then if you're, let me see real quick. So if you're saying your A band goes, that means that's how far the overlap is,
Oh, okay.
right? And these are your actin.
Mm-hmm.
And this one's right, because this is where you only have myosin. It's the
Okay.
H.
So this is
What was that? So for every, is it for every 3 there's 2?
I mean, in reality, they're equal,
Okay.
right? In 3 dimensions,
Okay. Because they just keep going.
right? It just keeps, build it up, circle.
Okay. Okay, so the A band's from. So is the zone of overlap part of the H zone or just the A band?
The overlap is the A band
Okay.
plus the H zone.
Okay.
The H zone is where myosin's by itself. So you're basically getting there. Yes. Whoa. Okay.
H band. These are the actins. That's A band, H band, I band,
Wait, what does that say?
H band.
So the H band is where you only have myosin, no actin. So you see up there, like it's basically the middle part. So the H is just from.
Here?
Where's your actin?
Right here.
Oh, your actin only goes halfway though.
Halfway from which? So from Z to.
Starts at Z and it goes a little bit over halfway towards M. See how you can see it up there in like the yellow? That's the actin. And see how it just starts at one end and it kind of stops halfway.
So the myosin is the one that starts from M to mid up,
Mm-hmm.
and actin from Z to the
Right.
Okay.
And they do overlap like a little bit, like 30% or.
And that's what
And the only thing that goes all the way from one end to the other is your Titan that you have there.
and what's it called when they're
See, that's where I was confused. I'm like, what?
what is it called when they overlap a little bit? It's just myosin, right? If they overlap.
The myosin and the actin overlap.
So that's where myosin is at, right?
Well, myosin's there, but it's also here. So like your myosin's here all the way to there. But no, it doesn't have its own name. If that's what you're
Is
thinking.
that
Of.
where the A band is at?
So the A band includes all the myosin. So it's the part that doesn't overlap and the part that does.
Okay.
So that'd be your A band.
And I band is only where actin is.
Right. You don't getting that, you think?
Is this right?
Where's your myosin? Which one's myosin?
What do you mean?
Well, on on C there, you drew your myosin. Did you label it?
Oh, they would be these, right?
They go all the way?
No, it's halfway, but then the titans, don't they overlap them?
The Titan's kind of next to it.
Okay.
And I know the book picture has it confusing, but all Titan does is connect M to Z.
Okay.
So it's the only one that
So
goes all the way.
Here's the M lines here, that's kind of hard to see.
What's that you're drawing?
Or the, not my myosin.
Well, myosin starts at the M line in the middle.
Okay.
I think I just got going a little too fast there.
Got it.
There's the,
I know, but I think you got going too fast in the beginning, and now
I don't know either.
Like, I don't
just erase this part,
Like none of
the one part,
all of it. I mean, keep the Z and the M,
Okay.
and then just start at C there. But start at C and really think about what is it telling you to do. Like, what is that you're drawing?
those ones are overlap.
So you got them, you think?
Yeah.
Yeah. Well, that's why we're drawing it, because you can show people that picture all day and no one knows what you're talking about. Right? It's like, what are all those things?
Wait a minute. So the Z disc is inside the inside the I-band?
So, I mean, you have it drawn right for now, but if you put a bunch of sarcomeres together, the next I band would be right here.
Oh, okay.
And then you get to the next
Oh, okay.
A band.
Oh, okay.
Yeah, it's like a rope. Mm-hmm. Sure. All right, y'all doing better?
I think so. I got it.
Where's your myosin? I would label them.
Let's say it's this one.
Mm-hmm. Yeah. Wait.
That's nice. Looks like
Yeah. Yeah. You're right. Pedro pointed at a different thing than you did.
Oh.
So you're right. Yeah.
acting is
So I would label them. So when you copy it down, you know what those lines are.
Can I just show you my paper?
Yeah.
That's for paper.
Well, I think I checked y'all's, right? So the only problem is the thin and the thick, they don't attach together like that. They overlap next to each other.
So,
So we could just draw like that, couldn't we?
Yeah, I mean, you could, but basically, I would move one of those lines a little bit above the other one, so like this.
so which one should I move above?
Well, like the thin and thick, they don't go together like that.
More like this.
They go like this when they meet. All right. So you got your Z's. This is, I know, that's why we're drawing it.
I'm gonna draw arrows.
There's a lot of extra space on the back if you want to draw it on the bottom half.
Yeah.
All right.
Okay. Now I know one's higher.
Okay.
I'm gonna steal this.
All right. And so then for C there, your myosin, you've got two that go halfway from here to here.
Okay.
That's your myosin. Same on your side.
For your myosin.
Mm-hmm.
Starts at the M line and goes halfway to the Z line. Yeah.
It looks better on the paper. I don't know. This looks better than what's on there. Well, I mean, so there should be something coming from the middle, coming out. So like your actin, I don't think you have your actin on there, right? Because it's right on here, right? It started here and went to overlap a little bit, but I don't see it on, or is it that? Oh, it is. There. There. It's missing. It's not attached. All right, so let me go ahead and draw that one in case anybody's still missing it. All right, so we got our Z's. There's another color maybe. All right, so in every sarcomere you've got your two Z lines on the ends, and then the M line in the middle. How many myosins did I have you draw? 2 or 3? All right. So this is myosin. And then the actin comes about halfway from the other side. This green one's working. We'll make ourselves a Christmas one. Same thing on the other side. And then our Titan comes from the Z to the M. We draw however you want. And then the different zones we're drawing. So the H zone is where you only have myosin. So it would start here and it would end basically here. That'd be the H. The A band's a little bigger. So the A band has all the myosin. So it would start let's get a little lower here, all the way to there. That's the A. And then anywhere where there's only actin, which is where the A band would end, that's the I. So I think most of y'all ended up with something like that. If you didn't, don't worry, it is pretty complicated. And of course, the reason to spend time looking at the structure is because anything else you do, if you don't understand how it's put together, none of the rest of it makes any sense. So the key thing to remember here is you've got the M line, that's what myosin's attached to. The Z disc, that's what actin's attached to. And then of course, it's not just one sarcomere in a cell, it's a whole long string of them. So then the next sarcomere would be exactly like this one, just right next to it. And then another one and another one, and so on and so forth. Any questions so far? No. Hold on, because you're going to redo it. I mean, you're going to do it contracted in a minute. All right, so let's talk about how that structure we drew, how it makes your muscles actually move. So it's really simple. Basically, the myosin, this red part here, grabs on actin and pulls on it. And kind of like this, like you're pulling on a rope, which we'll do in a second. And because actin's attached to the Z disc, if myosin pulls on all these ropes, what happens to these Z discs? They'll get closer together. And that's how your muscles actually make movement or contract, is these Z discs are getting pulled closer because myosin pulls on the actin. And basically they can keep pulling until the actin runs into the middle. That's how far they can contract. So myosin does the work, actin's like the rope attached to the Z disc. And myosin's the person pulling on it. And because all those Z discs are all attached together, and they're attached to the ends of the cells, and the ends of the cells are attached to each other, if you shorten all these sarcomeres, that means the whole cell shortens, and then the whole muscle shortens too, because it's all the cells are shortening at the same time. All right, so I think I won't make you redraw all of it. But next to your thing you already drew, why don't you draw it with contracted? But the only things you need to include, just put the Z, the M, the myosin, and the actin. So you don't need to draw all the bands or anything. So number 2, we want to see what it looks like if it was contracted, like the myosin pulled on the actin. Let me put this one back up here.
I drew it on the wrong side.
I drew another one.
Oh wow.
So I need extra paper. Do y'all need any paper, extra paper?
Do we throw it on the table or do we?
You can do it on the table first if you want. That's 2, but whatever.
Actually, 3.
So comparing it to what you already drew, which everybody erased. You weren't the only ones.
Do not erase it, sit on there too.
Are you serious?
Yes.
I should have put it on the paper. I put it up there.
Oh, up there.
I'll put it on the paper next time. So you think there's going to be more or less overlap when it's contracted?
They're gonna more overlap.
Okay. They don't look very overlapped to me there.
Well, that's because you threw it over there.
Well, that's not the contracted one.
Oh. So it'd kind of be like
So
this.
they have to be really tight.
You're right.
So tight. Like,
Not so much that they're closer up and down, is that they overlap more. So they're like this, and now they're like this.
so like that?
So like they started here, right?
Like that?
And now they're like that.
Just move them. Like, just draw them over. It looks like a hole. Looks like just a
Yeah, there should be space between them.
There you go, like that.
So for example, like how yours are barely touching right now, when they came close together, it should all be squished. Right? Because that's a good one. That's a good relaxed one. So then how do they look contracted?
So do they like go together? Do they bind together or do they like overlap?
They'd overlap more.
too much overlapping.
I think I would show the space there because otherwise you can't really tell what's what. Although you're right, technically they are touching each other.
So from like everything that
Jeremy, can I
overlaps.
have the marker?
Yeah, they're gonna overlap more than they did the first time. Are y'all writing these down somewhere?
Yeah.
Okay. Y'all are too? Oh, okay. Just for studying purposes.
I took a picture, I swear.
What are you thinking so far?
So that'd be.
Keep trying. You can do it. You can do it.
It'd be a lot. Like this will be farther towards the middle now, right?
Uh huh.
Then a
But
little
the
bit
actin.
there, Yeah.
Uh huh.
But the Titan stays the same.
Yeah, the Titan's still gonna. It's gonna get like squished like a spring,
More too.
but
So
I'm
the
not really
Z-disc
expecting it to-
come in, right?
mhm.
So
the
is that
whole
what it's supposed
thing-
to look like?
Well, kind of, yeah. But I want you to get the idea of it rather than just copy a picture down. So go back to your picture that you drew because it was good, right?
But wouldn't everything be shorter?
Uh huh. Right? Because you basically took this myosin, grabbed on this rope, and pulled it this way. And these are attached to here, so everything moves in.
If for me,
That's all that's
So we
happening.
draw it.
So our Z-discs are here, right?
More overlap. And so, like, you know, instead of the Z disc being here, maybe it's here and here now. That's all you're doing. Yeah.
Put it on here, is there? They're just,
I think I did mine wrong.
overlapping more.
Right. So the Z's came closer together.
Yeah.
So like if you went from here to there. Yeah, exactly.
Like that. Where's mine backwards?
I don't think it's what's backwards about it.
Because her myosin's longer. And my actin's longer.
I mean, it doesn't really matter because you've got the same
Either
idea.
one, they still go
Because they pulled the Z disc closer.
Right. Right.
I wrote it. Okay.
So yeah, what's the?
I mean, I would say compared to the first one, the myosin and the actin, they don't change the length. Do you know what I mean?
These are the ones that move.
They just go closer
Right.
together. Yeah, because
They
mine,
just like rub close together.
Mm-hmm.
see mine are like, they're on different lengths, they just.
Right. All right, so you got it all squished together. Makes sense, right? So it's like that looks good.
It's just like
This
drawn
way
out.
he had a, I just don't, I mean,
I don't understand it like that either. I understand it like that better.
I can tell my way through.
Like seeing it that way.
Do they tell the myosin and the actin?
Well, I guess the only way to know if it's right is to compare it to the first thing you wrote. Do you know what I mean? But yeah, all your actin and myosin are in the right place
These on the top are in the middle, and these are further apart. And when it's in fact it's closer together.
because it pulls on it. Right. Got anything? So it's all like squished. Basically, the Z discs are closer. There's more overlap. And you're right, like your actin here is almost touching the middle. So it got pulled over there.
Kind of like your fingers, like
Mm-hmm.
that's like extended and that's contracted.
Yeah. That'd be a good way to do it. All right, so I got it.
Yep.
It's looking good. Looking good.
Track it.
Do you
Alright,
have this one on just like? I'm just gonna print it out.
I can put it though.
You just wanna like redo that.
All right, shall I get them, you think? What's the 3 lines in the middle about?
so I think I draw one more step. Probably it's just the myosin and the actin right here. So like, oh, this is the M line.
Yeah, there's only one M line though,
So, okay.
right? So yours is right.
Better like.
I don't know why I'm not.
Here, take a look at this is here.
Okay.
So like the Z's got closer together. There's more overlap in the myosin and the actin. It's exactly what you want right there. So you got it. Good. All right.
So is there anything else going on? Making tracks.
Nope, that's it.
So this goes from
The Titan's getting squished.
halfway, so the then filament goes from being halfway over the thick to
all the way.
and so this part's still the thick filament too, right? Even though
Yeah, these are actually where the heads are, but
oh
that's where the pullers are. All right. All right, so you can see that up here, sort of what you were doing. So I think you already got a good answer there. Here's uncontracted, and you can see how far apart these two Z disks are, right, from here to here. And then down here, this is what happens fully contracted. So we moved all these Z disks closer together because we pulled on the ropes attached to them, which are the actin, basically. Do I? Yeah, so when it's contracted, if you look at this, the myosin and actin are basically completely overlapped. So in maximally contracted muscle, yes. Actin would touch the M, myosin touches the Z, basically. You like the gray one?
Yeah, it makes so much more sense with the I band and A band.
Oh, okay. I hate the gray one. But I'm glad we have both.
What part it really is.
Yeah, and now, of course, the reason they got all these names is way before we knew about what any of this stuff was. You could stain it and see, you could see this under the microscope, right? You saw these bands yesterday, at least some of them, right? You see kind of a dark and a light. That's what you were seeing. So they just gave them names because they didn't know what they were, really. Let's just call them A and I. That's why the letters don't really make any sense, right? Like H zone, what does that stand for? All right, so skip a few of these. I think y'all pretty much got it. Oh, we can do this one. All right, so I want to know which of those narrows or disappears when you're fully contracted. Let me put the answers up here, though. Answer choices. Where's the black one? All right. So A, H. All right. So which of those disappears when this is an uncontracted muscle? When it contracts, what goes away? H, I, M, or A band? All right, so why don't you talk to your group there and then we'll vote again. Wait, did I forget an answer? Oh, I need 2 more answers up there. Whoops. Sorry, I didn't put all the answer choices. Oh, I didn't realize it was 4. All right, 6 answers. Now you can choose what you want. All right, so mostly people put E, which is correct. So if you look back at this one, if you squish them all together, what are you left with? Basically only overlap. And the overlap, do you remember what the overlap was? Yeah, the overlap is the A band. So basically the A band includes all of the myosin, whereas the H zone is the myosin by itself. So if you don't have any overlap, you don't have any H zone anymore, and you don't have any I band anymore. Right. Let me do, yeah, all right. So another question. So which of these is true during contraction? All right, so chat with your group there. And if you do think it's... Do it? Oh, if you choose F, make sure you know which ones you think it is. All right, so I'll tell you that there's only one up there that is not true. So vote for the one you think is not true. So there's 3 that are true, 1 that's false. Or wait, how many are there? 5. 4 that are true, 1 that are false. 1 that's false. Choose the one that's not true. All right, so your time between, so most everybody's choosing C, which is correct. So if sarcomeres get shorter, Z discs come closer together, and then that's transmitted all through all the levels of the muscle. So muscle cell, myofibril, fascicles, the muscle. The tendon's the only part that doesn't change. But the muscle does, which is going to pull, on the tendon, which pulls on the bone. So they did get shorter to get you there. So for example, when I was holding this out, I had to get them shorter to get up here. But once I'm there, they're just staying contracted. Right, they're just staying short. It's a good question. And it's a good point to your isotonic, it had to start somewhere. It didn't just all of a sudden be there. All right, so we'll do all the, I think I'll just do all about the myosin cycle next time. And the types of muscle and exercise, you'll just have to learn that one from these slides. It's pretty simple, but this contraction we'll definitely go over together. It's complicated. Uh-oh.